I welcome you all to another session on this page, Knowledge Without the Price. And if you have not subscribed to this page, I don't know what you are doing. Trust me, you are missing out on a lot because this page has as its main objective purely health. On our platforms on Facebook, TikTok, YouTube, Instagram, what we focus on is health. And this page has two main branches. Now, the first branch is for healthy living and lifestyle. Are you currently being exposed to a particular infection and you don't know how, maybe an STI, maybe any sort of infection, and you don't know how to stay healthy? Trust me, visiting our page regularly will give you a lot of knowledge on how to live your life in such a way that you are not regularly exposed to those infections. So if you are not subscribed to our page, I don't know what you are waiting for. Now, the next branch of our page focuses on health, but this branch is for health enthusiasts, especially those into the field of laboratory sciences, because we dish out courses in the field of medical laboratory sciences, and we have already started. We started with biochemistry, medical biochemistry, wherein we looked at an introduction. We have looked at vitamins already, and today we are still to look at a very interesting topic. So if you have not subscribed to this page, trust me, you need to, because this page will give you a lot of knowledge when it comes to healthy living and lifestyle. So if you have not subscribed, I encourage you to subscribe and visit our other pages on Facebook, on TikTok, on YouTube, and on Instagram to get up-to-date knowledge with everything with regards to health and how to stay healthy. So for today, we'll be looking at another interesting lesson. In our previous class, we were talking about vitamins, and I explained extensively what vitamins are, their sources, and most importantly, I talked about their deficiencies, because there are certain things that people often experience. Because even in the lab, there are certain cases you see from patients, and you may be thinking, so far, meanwhile, it may just be a deficiency. So I explained everything with regards to vitamins, their deficiency, their sources. So that is the link. You can click the link on the channel or in this video. You go back and watch that lesson, and don't forget to jot down notes and pose your questions if there are any. So for today, we're talking now on another interesting lesson, enzymes. And those who follow this lesson to the end will have a special package from Knowledge Without a Price. So stay tuned and grab your pen, grab your notebook, because it's going to get serious now. Okay, so let's begin with enzymes. Now, majority of us, or most of us, or everyone from elementary knowledge, we have been hearing about the word catalyst. And in order to talk about catalyst, we can't talk about catalyst without talking about reactions. So if we talk about catalysts, we'll be talking about reactions. In the field of chemistry, physics, we'll talk about catalysts. There must be a reaction in view. For example, chemistry, there are some reactions which some compounds are being placed into that reaction, not because of the end products, but just because they need to speed up the reaction. And that is the same thing that happens in the body. There are certain compounds, or proteinous molecules rather, which aids to speed up chemical reactions, and they are called enzymes. So in summary, enzymes are biological catalysts which speeds up the rate of chemical reactions in the body and remain chemically unchanged at the end of the reactions. Note, they remain chemically unchanged at the end of the reaction. So before we proceed, let's look at the type of reactions that happen in the body. So there are predominantly two types of reactions that happen in the body. We have the anabolic and the catabolic reactions. Now we'll talk about the anabolic reactions. They are reactions which the main aim is to build up, while the catabolic are reactions which the main aim is to break down. So without much ado, let's look at the nature of enzymes. From previous knowledge or elementary knowledge, we were taught or told that enzymes are proteinous in nature. But trust me, that knowledge after this lesson, it will be shifted, because the aim of this class is to give us more knowledge and help us to be up to date with recent researches and recent advances that have been made in particular fields. So we'll be looking at those advances that have been made in the field of, or in that we made with respect to enzymes. So the first type of enzyme we look at is the simple enzyme. So the simple enzyme is the enzyme which has just its protein molecule. So from this statement, you can clearly depict that there are some enzymes which have non-protein molecules, and it opposes what we taught before, because we thought earlier we've been told that enzymes are proteinous in nature, but there are some enzymes which also contains non-proteinous molecules. So the simple enzyme contains just the proteinous molecule and it is not affiliated to any other molecule. And the next class of enzyme we'll look at is the holoenzyme. Now the holoenzyme is an enzyme which has both the proteinous and the non-proteinous molecule. So the proteinous molecule of this holoenzyme is called the apoenzyme, while the non-proteinous molecule is called the cofactor. Now if this cofactor is organic in nature, it is called a coenzyme. If it is non-organic, it is called an activator. And if this activator binds so tightly to the apoenzyme that removing the activator means denaturing or destroying the enzyme, it is called a prosthetic group. So in summary, enzymes are of two categories. You have the simple and the holo. Simple is just protein, while the holo is a proteinous plus non-proteinous. So the next thing we're looking at is the coenzymes. Now back to our lesson on vitamins. I explained extensively how vitamins aid or play a role in enzymatic reactions. So I encourage you to go back and watch or attend that class. So let's look at coenzymes. So coenzymes are derivative of vitamins. I explained earlier that talking about coenzymes that they are that happens when the cofactor is made up of organic molecules. So coenzymes are derived from vitamins, and now coenzymes binds directly to the active side of the enzyme. Now here is the role of coenzymes. The first thing, coenzymes aid in the transfer of functional groups from and to the substrate. And the next thing is that the same conformational change that happens in the substrate is the same thing that happens with the coenzyme, but the difference is that once the substrate is being translated into products, the coenzyme goes back into its original shape. And one important thing you have to note is that without the coenzyme, the enzyme won't function. So that is what you have to take note of about the coenzyme. The next thing we're talking about is the activator. Now we'll talk about activator. I made mention of the fact that they are inorganic molecules. So we'll talk about activator. We'll look at calcium, we'll look at magnesium, we'll look at copper, and we'll look at iron. Now as opposed to coenzymes, activators don't have anything to do with the active side of the molecule, but rather what activators do is that they bind to another side of the enzyme other than the active side, but activate or stimulate the enzyme. Now when we talk about activation or stimulating the enzyme, we're talking about adjustment of the adjustment of the amino acids at the level of the active side, to make it receptive for the substrate. And one thing that co-activators also do is that they promote the enzyme substrate complex bonding. We'll talk about this more as we proceed in the lesson. So it will be confusing because someone may ask, what is the difference between an activator and a coenzyme? One thing you'll notice is that an activator has nothing to do with the reaction, while the coenzyme has everything to do with the reaction, because the coenzyme binds directly to the active side of the enzyme and it is involved with the reaction. Now an activator is made up of inorganic molecules, while coenzymes are made up of organic molecules, specifically derivatives of vitamins. Now so that is what we have to take note of. Activators act as carriers, while coenzymes are not. They carry functional groups to and from the substrate. And as I explained earlier, when it comes to the active side, activators are involved because the same conformational change that takes place in the substrate is the same thing that takes place with the coenzyme, but the coenzyme is not translated, even though the substrate is translated into product. So in summary, coenzymes organic in nature, activators non-organic. Coenzymes, they aid in the reaction, they have a greater role to play in the reaction, while activators don't. So the next thing we're looking at now are the properties of enzymes. Now the first part of an enzyme we're going to talk about is the active side of that enzyme. Now all enzymes have active side. From elementary knowledge, they drew certain structures of enzymes and they put one small position in the middle, and they made us believe, oh, that's the enzyme, that's the active side. But that is true, but that is not how enzymes looks like, because the enzymes are just layers of amino acids which are being grouped together to aid or speed up the rate of reaction. And one thing we have to take note of is that one enzyme possesses multiple active sites, as opposed to what we've been taught for the purpose of understanding, not that that's how it actually looks like. One enzyme has more than one active site, which aids in the binding of molecule. Now here are the events which take place at the active site. I've talked about the coenzyme, and the activator. Let's see how they play a role when it comes to enzymatic reactions. Now, the active site is not just a protrusion, but the active site is a three-dimensional conformational layers of amino acids that we have to take note of. Now, the activator is going to stimulate the enzyme to be receptive to the substrate. Now, once the substrate fits into the active side of the enzyme, don't forget that the active side of the enzyme needs to be complementary to that of the substrate, making it receptive. Now, once it fits into that structure, the activator still is going to promote the bonding between the two, forming the enzyme substrate complex. Now, the coenzyme takes it up from here. What the coenzyme does is that it carries functional group from and to the substrate, causing diverse conformational changes. But once the substrate is translated into products, forming the enzyme product complex, the coenzyme goes back to its original nature, and the product dissociates from the enzyme and is being used up by the body dependent on the need. That is what happens at the level of the active side of the enzyme. Now, let's talk a little bit about the specificity of an enzyme. I made mention of the fact that the active side of the enzyme needs to be complementary. That's to tell you that active sites of enzymes are specific to the substrate they work on. Now, talk about specificity. There are two types of specificity. We have the absolute specificity and we have the stereo specificity. Now, we'll talk about absolute specificity. We are talking about the ability of an enzyme to be specific to a particular substrate. The ability of an enzyme to be specific to a particular substrate. Like, for example, look at the amylase enzyme, which is specific for starch to maltose. Now, let's look at the next type of specificity, that is stereo specificity. Now, we'll talk about stereo specificity as opposed to absolute specificity. Stereo specificity talks about the ability of an enzyme to be specific to a particular stereoisomer. For example, we look at glucose oxidase, which is specific to beta dextrose glucose, not alpha dextrose glucose. Now, if you want to know about what I just mentioned about the dextrose glucose, why they are not calling it glucose, and how come we have alpha and we have beta, I would like you to follow our notes. I would love for you to follow our lesson on carbohydrates, still on this page, and the link is down there on your screen. You can just click on it and follow that class after this one, so that you gain more knowledge. As I told you, we are interested in educating people about health. Now, the next property of enzymes is zymogenes. Now, we'll talk about zymogenes. I'll explain later as we proceed with this lesson. Now, talk about zymogenes. Zymogenes is the production of enzymes in an inactive form, which requires specific conditions to be active. Now, the production of enzymes in an inactive form that requires specific conditions to be active. For example, pepsinogen, which requires the low pH of hydrochloric acid to be activated to pepsin, which converts proteins to peptones. And lastly, we'll look at regulation, properties of enzyme. Now, the regulation of enzyme is one property of enzyme which is controlled by the body depending on the need, and it talks about the usage of activators, inhibitors, and effectors, and we are going to see the role this plays as we proceed with the lesson. So the next thing we're looking at is the classification of enzymes. Enzymes have different categories and classes, which aids for their names and their functions. So the first class of enzyme we're looking at is the oxidoreductase enzymes, and these are enzymes which catalyze the oxidation and reduction reactions, which we'll be talking about accepting and donating, or reception and donation of electrons. Now, another class of enzymes, these are enzymes which are involved in the transfer of functional groups from one molecule to the other, and they are called the transferase enzymes. Now, another class of enzymes are enzymes also which are involved, which catalyze reactions by breaking down, by adding water, which catalyzes reaction by adding water, because catalysis is only breaking down. So which catalyzes reaction by adding water, called the hydrolysis enzymes. While the lysis enzymes are enzymes which catalyzes reactions without adding water nor adding electrons. And the isomerase enzymes, these are enzymes which rearrange the structures of molecules with respect to their isomers. And the ligase enzymes, these are enzymes which join two molecules together. This is just an overview of how enzymes are classified. Now, if you want an intensive classification lecture, or if you want to explain in details, make sure you leave a comment and I'll do that lesson for you. Now the next thing we're looking at is the mechanism of enzyme action. As I've already explained that with the active side, but I'll just repeat it again due to this particular topic that we are talking about. Mechanism of enzyme action. Now, one thing we need to note is that we'll talk about an enzyme binding to the substrate. We are not referring to the whole enzyme, rather, we are talking about the active side of that enzyme binding to the substrate. And for an enzyme to bind to a substrate, the active side of that enzyme needs to be complementary. It means that now we look at the lock and key hypothesis, which states that the enzyme is the lock, while the enzyme is the key. The enzyme is the lock, while the substrate is the key. And for the reaction to occur, they must be complementary. That is, one should be able to fit perfectly into another. And what promotes that bonding is called the activator. As I explained earlier, it stimulates the enzyme, making the structure of the amino acid at the level of the active site receptive. Now, once that bond has been initiated, the coenzyme takes it off from there. It aids in the transfer of functional group to and from the substrate, leading to the production of the enzyme product complex. Don't forget, once the substrate binds to the enzyme, the formation of the enzyme substrate complex aided by the activator. Now, since as the conformational changes have already occurred, there will be the formation of the enzyme product complex, which will be aided now by the coenzyme. And once the product is being formed, it leaves the enzyme and is being used up by the body dependent on the need, while the coenzyme goes back to its original shape. So that is what we need to take note of when it comes to the mechanism of enzyme action. The next thing we're talking about is factors affecting enzyme activity. We look at three factors: temperature, pH, substrate concentration. Now we'll talk about temperature. Here's what you need to note. At low temperatures, enzymes are inactive. At high temperatures, enzymes are denatured. And the best temperature for enzyme to work is called the optimum temperature. As simple as that. And what you have to note of is that in knowledge without a price, in this school, as I will put it, we simplify knowledge a lot. We make sure it can be comprehended without stress. And we have it as our logo. The applied knowledge is power, not necessarily knowledge, but the one which we are able to apply is what makes the difference. Now, the next thing we're talking about is the effect of pH. Now, one thing we need to take note of is that enzymes are denatured at low pH, but it doesn't work for all enzymes. Some enzymes work best at low pH because the optimum temperature, which is the best temperature, the optimum pH, I'm saying we're talking about temperature, we're talking about pH now. The optimum pH, which is the pH which enzymes work best, is the neutral pH. Now, the low pH is expected to denature enzymes, while high pH is expected to make them inactive. But some enzymes have certain exceptions, because an enzyme like pepsinogen works best in low pH. So that is what you have to take note of. But what you have to note is that low pH is going to denature enzyme. High pH is going to inactivate them. And neutral pH is the best pH for most enzymes to work, but there are exceptions. That's what you have to take note of in summary. Now, the last factor that we'll look at is concentration of the substrate. Now, we all know that, or what you have to take note is that one enzyme has more than one active site. That's multiple active sites, which makes it receptive for many substrate molecules. So as you increase substrate concentration, the velocity of enzyme reaction also increases. But once you exceed the enzyme's maximum capacity, the speed of the reaction begins to drop, which slows down enzymatic reaction or the products from enzymatic reaction. And that is how substrate concentration affects enzymatic reaction. So the next thing we're going to talk about now is enzyme inhibition. Remember when I was talking about regulation, I made mention of the fact that with time, or in the course of the lesson, I'm going to explain what inhibition means. So we'll talk about enzyme inhibition. We are looking at any molecule that slows down or stops the velocity at which enzymes catabolizes or anabolizes, or enzymes catalyze reactions. So in simple terms, that is an inhibition. A molecule that stops or slows down the rate at which enzymes catalyzes reaction. And now the first type of inhibitor or inhibition we'll look at is the irreversible inhibition. Now we'll talk about irreversible inhibition. We are looking at molecules which cannot be removed from the enzymes by adding the substrate. Now, this is a molecule which binds completely to the active side of the enzyme and prevents substrates from binding. Now, so it cannot be removed by increasing substrate concentration. This is to tell you that there are some inhibitors that can be removed by increasing substrate concentration. The next type of inhibitor we're talking about is the reversible inhibitor. Now we'll talk about the reversible inhibitor. There are two types. We have the competitive and the non-competitive. Let's look at what the competitive inhibitor means. Now, from its name competitive, it competes with the active side of the enzyme. It competes with the substrate, for active side of the enzyme, as I said earlier. Now, in this type of inhibition, there are two types of reaction that occurs, because since as there is a competition, the inhibitor binds to some of the active side, while the substrate binds to some of the active side, as opposed to irreversible, wherein there is no binding of the substrate. Okay, so that's what I've taken note of. Now, there are two types of reaction that happens in this case. You have the enzyme inhibitor complex, that's the first type of reaction, and you have the enzyme substrate complex. So once the inhibitor binds to the active side of the enzyme, it forms the enzyme inhibitor complex, which leads to no product from slowing down enzymatic reactions, wherein when the substrate binds to the enzyme, it leads to the enzyme substrate complex, and it leads to products through the processes I explained earlier in the lesson. If you want to know about the processes, you can go back and listen to it when I was talking about mechanism of enzyme action. So that is what you have to take note of. The inhibitor binds to the active side of the enzyme, and the substrate binds. There are two types of reaction that will be formed: enzyme inhibitor complex and enzyme substrate complex. The enzyme substrate complex leads to the release of products, while the enzyme inhibitor complex leads to no products, slowing down enzymatic reaction. The next type of inhibitor we're talking about is the non-competitive inhibitor. Now, the non-competitive inhibitor doesn't bind to the active side of the enzyme, but bind to another side. Likewise, the effector, but what it does is that it changes the conformational structure of the active side, making the active side unreceptive for substrate. Now, as activator makes the active side receptive, non-competitive inhibitor makes the active side unreceptive. So it's difficult for substrate to bind. So you see that as opposed to the competitive inhibitor, the non-competitive inhibitor is worse, because during competitive inhibition, there is a probability that you will still have products formed, but with the non-competitive inhibitor, no product will be formed. So the next thing we're talking about is regulation of enzyme activity. Regulation of enzyme activity. The first type of regulation we'll talk about is zymogen activation. Zymogen activation is the first type of regulation we'll talk about. Now, I mentioned earlier that we'll talk about zymogens. We are talking about enzymes that are produced in an inactive state but require specific conditions to be produced. And now this helps to regulate enzyme action, because the activation of this enzyme is dependent on the body's needs, and the inactivation of these enzymes also preserves tissues. So production of enzymes as zymogen helps to regulate enzyme action dependent on the body's needs. Now, if the body's needs is high, there will be certain mechanisms that will be produced that will activate the enzyme, and if the body's need is low, the enzyme will continually remain in an inactive state, preserving the surrounding tissues. Now, the next type of regulation we'll look at is allosteric modulation, and this allosteric modulation is strictly for simple enzymes. What I've been explaining earlier works for hollow enzymes, but allosteric modulation works strictly for simple enzymes, because simple enzymes just bind to substrate and inhibitors. They have no coenzymes, they have no activators. They bind just to substrate and inhibitors. Now, we'll talk about allosteric modulators or allosteric enzymes. We'll talk about the ability of enzymes, enzyme actions to be regulated dependent on allosteric effectors. Because for allosteric modulation to occur, certain molecules are being produced called effectors, which the enzyme binds to. Now, when we talk about the effectors, there are two types of effectors. You have the positive and the negative effectors. Now, once the enzyme binds to the positive effector, once the enzyme binds to the positive effector, there will be an increase in the rate at which enzymatic reactions occur. But once the enzyme binds to a negative effector, there will be a drastic decrease in the rate at which enzymatic reactions occur. So that's what you have to take note of about allosteric modulation, the ability of simple enzyme to bind to specific molecules called effectors. which regulate or modulate their actions. So that said, we have come to the end of this lecture. As I mentioned earlier, that there is a special package. So Knowledge Without a Price is going to post a quiz or is going to post series of questions from this specific lesson with our email on it. So I expect every student to answer the questions based on this lesson, or answer the question based on what you have understood from this lesson, and submit your answers to the email. It will be corrected and sent back to you through email. Don't forget to like, don't forget to share, don't forget to subscribe. Follow all our social media handles on Facebook, YouTube, TikTok, Instagram, so as to be up to date with the knowledge we are going to release based on either on health knowledge or healthcare or lifestyle management and metabolism. Don't forget to like, share, and subscribe. Thank you.